And today we are going to discuss about the concentration and dilution processes of the urine. And in this lecture we are going to discuss about a bunch of details regarding this process. So first thing is first, when a person is intaking a large amount of water, their body fluid starts to become hypoosmolar. And you know that normal osmolarity of the blood and the body fluids is approximately 290 milliosmol per liter. Okay, it's the standard. And if the person is ingesting large amount of water, so this blood osmolarity starts to decrease. It means the blood starts to become hypoosmolar, and we cannot afford that, right? It's very dangerous. So one of the major functions of the kidney is osmoregulation. It maintains the osmolarity of the body fluids by adjusting the urine osmolarity. And this process is so efficient that even 1% change in the body fluid osmolarity changes the urine osmolarity. And for your information, that a normal male about 70 kilogram produces 600 milliosmol of solutes as waste products per day, and these need to be excreted out of the body. And when a person is ingesting a large water intake, it means that he is going to produce large amount of diluted urine, right? There is no debate about it. And when there is decreased or no water intake, it means that the person is still losing the water by respiration and sweating, and there is the danger for the concentration of the body fluids. It means that osmolarity can be increased from 300 maybe to 310, maybe to 320. Okay, there is the danger for hyperosmolar conditions. It's also very dangerous. So person will pass small amount of concentrated urine because we want to conserve water in case of no water intake. So let's talk about that, how the kidney maintains the urine concentration and dilution. First of all, kidney require two things for the maintenance of this urine osmolarity and for the osmoregulatory functions. First is the renal medullary interstitial fluid must be hypo or hyperosmolar. What does it mean? It means that when we move from cortex to medulla, the osmolarity of the interstitial fluid increases progressively. Okay, the medullary interstitial fluid is hyperosmolar. So there is cortico-papillary osmotic gradient present in the nephron. And the second is antidiuretic hormone has a role. We will discuss them. So for your information, that when a urine osmolarity increases more than 300, the urine is hyperosmolar. When it's decreased less than 300, it means that the urine is hypoosmolar, and when it is equal to 300, it means the urine osmolarity is isoosmolar urine. We have discussed that cortico-papillary osmotic gradient is important, but how to produce cortico-papillary osmotic gradient? First of all, okay, so let's produce the cortico-papillary osmotic gradient. We require two things. First of all, we require loop of Henle as countercurrent multipliers, and second is the urea recycling in the renal medullary area. And once the corticopapillary osmotic gradient has been produced, then how to maintain that? For the maintenance purposes, we have specialized capillaries known as vasa recta as countercurrent exchangers. I will explain these all of the terminologies one by one. First of all, let's see that how corticopapillary osmotic gradient is created. When the tubular fluid is filtered, when the blood is filtered, okay, from the glomerular capillaries, the blood starts to reabsorb first in the proximal convoluted tubule. And you know that in the proximal convoluted tubule, the reabsorption of water along with the other ions like sodium chloride, glucose, etc. is isoosmolar. It means that if 65% of sodium is reabsorbed, then 65% of water will follow. So what would you expect that at the end of proximal convoluted tubule, do the osmolarity change? No. Osmolarity of the tubular fluid does not change because the reabsorption of ions along with the water in the proximal convoluted tubule is isoosmolar. It does not changes the osmolarity of the tubular fluid. But when the tubular fluid descends down the descending loop of Henle, okay, descending part of loop of Henle, as you know that there is cortico-papillary osmotic gradient. For example, here the osmolarity is 300 in the interstitium. Then when starting from the cortex towards the medulla, the osmolarity starts to increase. Eventually it becomes 1200 at the medullary interstitial fluid. And when the blood is passing towards the descending part of loop of Henle, as you know that this part is permeable to water and solutes. When this tubular fluid notice that outside the interstitium is hyperosmolar, so it starts to lose water. Okay, that's logical. The water moved from low concentration to high concentration of solutes. So eventually the tubular fluid gained osmolarity because it has lost the water. It has lost the water. So the tubular fluid osmolarity starts to increase in the descending limb of loop of Henle. But when the tubular fluid comes in the ascending part, there is a special transporter known as the sodium potassium chloride 2 chloride cotransporter. It's sodium potassium 2 chloride cotransporter on the luminal membrane. What the function of the sodium potassium 2 chloride cotransporter? It transports solutes from the lumen into the cell and from the cell out into the interstitium, and it contributes to the hyperosmolarity of the medullary interstitium because a lot of solutes are present here. A lot of solutes have been pumped from the lumen into the cell and from the cell into the interstitium. Eventually, the tubular fluid starts to become hypoosmolar, okay, because the solutes has been washed away, and the interstitial fluid starts to become hyperosmolar. It means that the thick ascending part, to be more specific, contributes to the hyperosmolarity of the medullary interstitium. Okay, and when the tubular fluid leaves the thick ascending loop of Henle, the osmolarity of tubular fluid has been decreased. It is logical that all of the solutes, mostly solutes have been reabsorbed, so osmolarity automatically decreases. In the early part of the distal tubule, it is known as the cortical diluting segment. It means that it again reabsorbs some amount of solute and it again decreases the osmolarity of the tubular fluid approximately to 60. But there is a debate about the distal tubule, the cortical collecting tubule, and the medullary collecting tubule. So what do you think, that 60 will remain 60 or it will increase or decrease? Let me explain. That in the absence of ADH, when there is no ADH, no antidiuretic hormone, it means that this 60 will remain 60 because water has not been reabsorbed. But in case of the presence of antidiuretic hormone, water will be reabsorbed in the late part of nephron and this osmolarity starts to increase. You understand that it starts to increase because water has been pulled out and the solutes are concentrated in the tubular fluid, and we pass concentrated urine in small amount in the presence of ADH. So this was about the cortico-papillary osmotic gradient. But why I have said that loop of Henle work as a countercurrent multiplier? Because this countercurrent mechanism of the loop of Henle increases and multiplies the capability of the system to generate hyperosmolarity. That's why we call the loop of Henle as a countercurrent multiplier in generating the medullary hyperosmolarity. Do you see that if the nephron were straight, the ability to generate hyperosmolarity would have been approximately 400. But in reality, the nephrons in the medullary interstitial fluid have a hyperosmolarity of about 1200 milliosmol per liter. It is due to the curving of the loop of Henle. It's due to the loop of Henle. So loop of Henle, due to the loop of Henle, the ascending part and the descending part, the ability of the nephron to develop hyperosmolarity is multiplied. That's why we name them as countercurrent multiplier. Okay, as you know, and the first thing in the development of the cortico-papillary osmotic gradient was the countercurrent multiplier, that is the loop of Henle. What was the second thing? Second thing was urea recycling in the inner medullary collecting tubules, right? So as you know that this part of nephron from the PCT and the descending limb, this is water permeable. It means that water can be reabsorbed from here. And this part from the ascending thick part and the ascending thin part and the early distal tubule, this part of the nephron under all physiological circumstances is impermeable to water. Here water cannot be reabsorbed under physiological circumstances. And the late part of nephron, that is the late distal tubule, the cortical collecting tubule and the medullary collecting tubules, their water reabsorption depends on the function of antidiuretic hormone. Okay, if antidiuretic hormone is present, it will allow the water reabsorption, and if it is not there, water cannot be reabsorbed in the late part of nephron. The second thing was urea recycling mechanism, because the first was the countercurrent multiplier in producing the cortico-papillary osmotic gradient. It means that small amount of urea is reabsorbed in the proximal convoluted tubule. It is permeable to urea, but other part of nephron, these are impermeable to urea. They do not allow urea reabsorption. Only the last part of nephron, which is known as the inner medullary collecting tubule, allows the urea reabsorption, because this part has special transporters for urea. It means that they can allow urea to be reabsorbed, and when the tubular fluid reaches this part of the nephron, it is highly concentrated with urea, because urea has not been reabsorbed in any other part of the nephron, right, except the proximal convoluted tubule. Urea will enter the interstitial fluid and it will contribute for the medullary interstitial hyperosmolarity, because urea itself is a solute. So when a large amount of urea is present in the medullary interstitium, it will contribute to the hyperosmolar conditions of the medullary interstitium. But why we call this mechanism as urea recycling mechanism? Urea does not stay in the interstitium for a long time. It will again be taken up by the loop of Henle. Okay, it will taken again up by the loop of Henle and transported along the tubular fluid, and again it reaches to the inner medullary collecting tubules, and eventually it will be passed into the urine. Of course, not all of the urea is reabsorbed here. A lot of it is passed into the urine all the time, because urea is of course the composing the urine. Mostly 50 to 60% of the urine composition is urea. So that's why urea is passed into the urine and also is reabsorbed into the interstitium. So what we have understand? We have understand the cortico-papillary osmotic gradient is the term used because there is progressively increasing hyperosmolarity when we move from cortex towards the medulla in the nephron, and eventually the hyperosmolarity is approximately 1200 milliosmol per liter, and this is The development of the cortico-papillary osmotic gradient is due to the two things. First is the countercurrent multiplier, and second is the urea recycling mechanism. Countercurrent multiplier, we call them as the loop of Henle, because in the descending part the water is lost, because when the tubular fluid reaches the deep into the medulla, it recognizes that in our surrounding there is hyperosmolar interstitium, so this starts to lose water as the descending part is permeable to water. And in ascending parts, a lot of solutes has been reabsorbed and they are pushed into the medullary interstitium, contributing to the medullary hyperosmolarity. This mechanism multiplies the capability of the system to develop the cortico-papillary osmotic gradient. That's why we call them as countercurrent multiplier. Second is the urea recycling mechanism. In the last part of nephron known as the inner medullary collecting tubule, there are special transporters for the urea reabsorption, and they allow the urea to be moved into the interstitium, and this reabsorbed urea will contribute to the medullary hyperosmolarity unless it is reabsorbed and taken again by the loop of Henle, become a part of tubular fluid again, and this is a process by which the urea recycle occur in the nephron. It is very different from urea cycle, that is something in biochemistry, but now we are discussing about urea recycling mechanism, right? We have discussed this in detail, and in the next part of the urine dilution and concentration, um, we will discuss about the maintaining part, which was the vasa recta, and we will discuss about the effect or influence of antidiuretic hormone on the urine concentration. So this was for today. I hope you understand the video, and take care. I will see you in the next video. Till then, goodbye.